Hey everyone, so today we're going to be covering the metabolism of iron. Before we get started, I just want to go over the points that I'm going to be covering. First, we're going to discuss where we got our iron from. We have two sources. We're going to discuss the pathway of each source. We're also going to be discussing the regulation of metabolism. When we have too much iron, what does our body do to take care of that? When we have too little iron in our plasma, what does our body do to take care of that? We're also going to be going over some disorders. So when we have iron overload or iron deficiencies and what we see and how they manifest on lab tests. Okay. Again, before we get started, just like our human porphyrias lecture, I wanted to go over just a few points just to make everything clear. We have two forms of iron that we are concerned with as human beings. We have ferrous iron, Fe2+, or heme iron, and we have ferric iron, Fe3+, or non-heme iron. So from the names, we can infer that ferrous or heme iron is the one we need for heme and for hemoglobin. And if you reviewed heme and porphyrias, you'd also know that Fe2+ is required in the last step of heme biosynthesis. But we also need to know about ferric iron because we actually ingest iron as ferric iron. And we'll go into that in a bit. Also, we must know that ferrous iron, Fe2+, is the only type of iron that is absorbed in our duodenum. And again, it carries oxygen, whereas ferric iron does not. Hence, it's non-heme title. All right, so, here's just a silly way you can remember it. Fe2+ goes into the duodenum. Fe2+ carries O2. All right, so let's start with our main points. So there are two possible sources of iron. We have diet and recycling senescent red blood cells from macrophages. What does this mean? This means that when it's time for our red blood cells to basically pass on, they get super old, they go to our macrophages, take them in, break the hemoglobin into heme and globin, and then it's going to further break down heme into Fe2+ and biliverdin. Now, this we're not concerned with. This is what we are concerned with. The Fe2+ that we take from heme, so keep in mind we're still in the macrophage here, it's going to go through a portal or a protein called the ferroportin or the ferroporta protein. And this ferroportin protein is often accompanied by fastin, which is basically just a peroxidase with ceruloplasmin. And what this does is that it oxidizes Fe2+ into Fe3+. And you could probably guess from the name, ferro meaning iron, oxidizes. All right, so this Fe3+ is going to be released into our blood. However, it can't just roam freely like that because it can cause lots of issues, which we'll get into later on, more specifically the Fenton reaction. It's going to be bound to a protein called the transferrin protein and then stored in tissue stores. And this is what's going to happen if we were to get our iron from senescent red blood cells via macrophages. So let's move into the more complicated source, which is diet. So if we get, let's say you have a bowl of spinach stew or something that's super rich in iron. The iron that's going to be in your food is going to be ferric iron or Fe3+. And we know that Fe3+ cannot be absorbed into our duodenum and it cannot carry oxygen. So we need to change that. We need to change that to ferrous iron. And how does that happen? So once you ingest it, it gets to your stomach, obviously. And in your stomach, there are two agents or factors, if you will. There are two conditions that have to be met in order for us to get to ferrous iron. We need gastric acid, HCl, and vitamin C. All right, moving on. So we've got our ferrous iron. Now this ferrous iron is going to move into the duodenum, and we said that ferrous iron, Fe2 plus, moves into the duodenum. Once it gets to the duodenum, actually, what cells do we have there? We have our enterocytes. Ferrous iron is going to be absorbed into our enterocytes. So how does that happen? We actually need to know a few things about our enterocytes. Well, first of all, again, only Fe2 plus ferrous iron can be absorbed. And this happens via one of two channels, the heme carrier protein channel or our divalent metal transporter channel or our DMT 1. Okay, so let me just draw a diagram here to make it easier to understand. So here's our heme carrier protein channel and our DMT1. Ferrous iron will enter through one of these channels, either this one or this one. And once it's inside the enterocyte, there's actually two possible things that could happen. It's either stored or it is brought back into the blood as ferric iron. So let's focus on this one for now. It's stored as mucosal ferritin. So what's ferritin? So what is ferritin? Ferritin is just a protein that takes up or releases ferrous iron. Here's a few points that you need to know about ferritin that were mentioned in your lecture slides. So I'm just going to be explaining each point right now. Before we start, I just want to mention that in men, you're supposed to have 800 milligrams, and in women, you're supposed to have 200 milligrams of ferritin. It's an acute phase reactant. Now, what does that mean? What is an acute phase reactant? An acute phase reactant, in simple terms, is just an indicator that something is wrong with your body functions. So if you have something that is an acute phase reactant, like ferritin in this case, let's say you have a patient come in and they have some really weird symptoms, and you do a lab test, and you get your lab test results, and they have really, really high amounts of ferritin. This high amount of ferritin is going to tell you that there's an inflammation going on. Now, when we have something that its increase indicates inflammation, this is what we call an acute phase reactant. It's just a fancy word for something that tells us that there is inflammation going on when it's in high amounts than usual. Okay, so that's what an acute phase reactant means, and ferritin is an acute phase reactant. Ferritin increases with liver inflammation, again, exactly what an acute phase reactant is, and it increases in anemia. Now, this might sound counterintuitive. How does ferritin, which is a protein that binds to iron, increased in anemia? Well, in your body, this person is anemic, okay? And in this person's body, and in all of our bodies, we usually have free iron, obviously in very small regulated amounts, but it's there nonetheless for a reason. We also have stored iron in the liver and the spleen. All right. So let's say we have an anemic person. So in an anemic person, they're going to be iron deficient. So to compensate for that, your body is going to go to these stores and mobilize them so that we have more iron. But when it does that as a compensatory mechanism, it overdoes it because it's in a panicked state, which is why eventually it's going to be elevated more than usual. Okay, so we've understood what ferritin is. So let's go back to what we were discussing. Again, our enterocytes have two options to deal with the iron that it receives. It can either store it as mucosal ferritin, or it could mobilize the ferrous iron, oxidize it into ferric iron, and then release it into the blood. So how does it do that exactly? So there's another protein that we need to know about called ferroportin protein. And if you'll recall, this is also in macrophages along with enterocytes. This is our enterocyte. And if, again, you recall, these ferroportin proteins have something called hephaestin, which is a peroxidase with ceruloplasmin that basically takes the Fe2 plus, oxidizes it into Fe3 plus, and then sends it out into the bloodstream. Now this ferric iron that is sent out into the bloodstream needs to be bound, because otherwise we're going to end up with Fenton reaction. So what is this Fenton reaction that we mentioned at the start as well? So the ferric iron, free ferric iron that is not bound, will cause a reaction that will end in the production of free radicals. Now, we usually have free radicals surrounding our cells, and these usually help us deal with foreign materials, but having too much of them is going to be dangerous for our healthy tissues, because they're going to start to attack our healthy tissues, which is why we need to make sure that we have enough transferrin. Transferrin is what binds ferric iron in the blood. So once ferric iron, Fe3 plus, is bound to this transferrin, again, from here on out, we have two options, two possible things. And this step is very important in terms of iron regulation, because whatever happens here kind of decides whether we stimulate hepcidin, we down-regulate iron absorption, or we go on to form red blood cells from erythroid precursors. So going for the bone marrow option, you're going to stimulate the erythroid precursors, which are stem cells that are destined to become red blood cells, to mature into actual red blood cells. But if this transferrin ferric iron complex chooses to go to the liver, what the liver is going to infer from this is that there is a lot of iron in the body right now, and we need to take care of it, and we need to downregulate how much iron is being absorbed into the body. This is when hepcidin comes into play. So what is hepcidin? Hepcidin is a regulator. It's a negative regulator of iron absorption. It takes care of both sources. It takes care of the dietary iron, and the iron that we get from senescent red blood cells. And we're going to be discussing those two mechanisms in more detail in a bit. So just a piece of information that we need to know about transferrin, which is, again, the protein that binds ferric iron in the blood. It is a negative acute phase reactant. Now, why is it a negative acute phase reactant? First of all, what is that? Let's just go over that again really quickly. An acute phase reactant is something whose increase indicates that there is inflammation going on. So a negative acute phase reactant would be the opposite of that. Its decrease would indicate that something is wrong. So why if we have decreased transferrin levels less than normal, does that tell us that there's inflammation going on? So let's say we have an inflammation going on. Inflammation is one of the things that stimulates hepcidin along with iron excess. And these two actually make sense if you think about it. If you have a lot of iron, you're going to be having a lot of iron that's going to be rushing to both the liver and the bone marrow, stimulating hepcidin. The second thing is inflammation. Inflammation stimulates hepcidin. So if we know that inflammation stimulates hepcidin, this means we're going to have lower iron absorption. If we have lower iron absorption, then this is not going to happen. And then we're not going to have the ferric iron in the first place to be bound to transferrin. So we're not going to need this transferrin, and it's going to be downregulated, which is why we're going to have lower transferrin levels in inflammation, making it a negative acute phase reactant. Over here is just a figure that was mentioned in your lecture slides that you might need to know. In this lecture, learning objectives were not stated, and there are quite a lot of figures and numbers that are listed that I can't help you with. They're just static figures that I guess you'll have to memorize in the case that you need to know them. And I've just included one of them here, which is the total iron binding capacity of transferrin, meaning how much iron can 1 unit or 1 deciliter of transferrin bind. It can bind 300 milligrams of iron. So for each 1 deciliter of transferrin, 300 milligrams of iron are bound. Okay, let's talk more about regulation. So we've discussed iron excess and how it increases hepcidin, which decreases iron absorption. We talked about inflammation. Let's talk about things that downregulate hepcidin. So there's 3 things. We have iron deficiency, hypoxia, and ineffective erythropoiesis. Iron deficiency is kind of makes sense, because if you don't have enough iron, you don't want to downregulate it even more. You need that iron to be absorbed, so you're not going to want any hepcidin. Hypoxia is a decrease in oxygen. If you have low oxygen, you want that to be absorbed so you can make more red blood cells. Ineffective erythropoiesis. So if your body's trying to make red blood cells, erythropoiesis is just a fancy word for red blood cell synthesis. If your body's trying to make red blood cells, but it's failing, this is going to tie back to a lower ability to carry out oxygenation functions, which ties back to hypoxia, which is, again, same reason why we don't want hepcidin. So how does hepcidin actually work? Again, we said that it tackles both sources, the dietary iron and the iron we get from senescent red blood cells. How does it do that? You'll recall that both enterocytes and macrophages have something in common. They both have the ferroportin receptor. Degradation of this receptor will cause reduced iron release from the macrophages and reduced duodenal iron absorption. So it's going to take care of both of these. So on the other hand, what if we have insufficient iron? We don't have enough iron that is reaching the liver. Again, iron deficiency. Hepcidin will be downregulated, allowing more iron to be absorbed. Now, how is it downregulated? When hepcidin needs to slow down its role a bit with downregulating iron, it's going to go to the ferroportin. These are both ferroportins, but this is on a macrophage, and this is on an enterocyte. And this is the hepcidin. It's going to go to that ferroportin, going to be taken in by endocytosis and then broken down. And then hepcidin is going to be downregulated. So let's talk about ceruloplasmin. Ceruloplasmin, it's found with the ferrooxidase proteins, hephaestin, that are usually located near ferroportins. This is because in a macrophage and in an enterocyte, in both of these, we have Fe2+ that's entering these cells, but we need them to exit in their Fe3+ states because they need to be bound to transferrin, because we need that transferrin ferric complex. which is why we have ceruloplasmin. Now, ceruloplasmin here oxidizes Fe2 plus to Fe3 plus to be released into the blood and bound to transferrin. So there's two clinical correlations here that were mentioned that I added. We have hypoceruloplasminemia and aceruloplasminemia. Hypo, let's break this word down, meaning low, ceruloplasmin, emia, meaning blood. So we have low ceruloplasmin in the blood. So in enterocytes and in macrophages. In enterocytes, this ferroportin, if it lacks ceruloplasmin, then it's not going to be able to pump out Fe3 plus into the blood, and Fe2 plus is just going to be accumulating, and it's not going to be able to get out. Again here, accumulation of iron in the liver and other tissues. Same thing for our macrophages. We have our senescent red blood cell here. It's trying to get rid of its iron, heme, Fe2 plus. It's not going to be able to get out as its Fe3 plus state because it's not getting oxidized because we don't have enough ceruloplasmin. So there's going to be inefficient iron recycling here. Okay. Our next condition is aceruloplasminemia. A meaning without, ceruloplasmin, blood. So let's say we have no ceruloplasmin at all, meaning no oxidation of ferrous iron. So we have no peroxidase activity. This is very bad because here you have some activity. Yes, it's not sufficient, but it's there. You have inefficient iron recycling, but here you have no iron recycling whatsoever. So it accumulates in the islet cells of Langerhans in the pancreas and the basal ganglia in your brain, which is how it causes type 1 diabetes if it accumulates in your pancreas, and neurological degeneration when it accumulates in your basal ganglia. Alright, so these are all the lab tests that you could perform if they have an issue with iron. You have a red blood cell count, which you can get a hemoglobin estimation from. That's number one. You can test for plasma iron levels, total iron binding capacity, and transferrin saturation percentage. You can also test plasma ferritin, a Prussian blue stain, and iron in tissue biopsy. So you could test how many micrograms of iron per 1 gram of tissue sample. Now, this Prussian blue stain is actually going to test for hemosiderosis. Hemosiderosis just means an accumulation of hemosiderin. Hemosiderin is basically just a less soluble form of ferritin, and its accumulation can be detected with a Prussian blue stain. So how could we end up with an iron deficiency? There's actually quite a couple of ways. You could either have an impaired protein. One of these proteins could be impaired, or more than one. Hemoglobin, aconitase, succinate dehydrogenase, or your hemoprotein, like your peroxidase and your cytochrome. But what if these proteins are perfectly normal? Well, then you could have a malabsorption problem. These proteins could be working perfectly fine, but when it gets to actually absorbing the iron, you could have a GI issue that's interfering with that, like celiac disease. Another reason why a patient might have iron deficiency is due to blood loss during menstruation. This is actually why women have higher iron requirements, because women lose more blood on average. And if a woman is coming in with an iron deficiency, it may be because she's not meeting her iron requirements and she's losing too much blood during menstruation. Another reason is GI bleeds. Again, any GI issue that may cause loss of blood, or even parasites, which can act as a mechanical barrier to iron. In pregnancies, you have higher iron requirements, meaning pregnant women are at a higher risk of experiencing iron deficiency. On the other hand, we could have iron overload. hemosiderosis. We discussed this, meaning you have accumulation of this form of ferritin that is detected with a Prussian blue stain of a biopsy. So we take a biopsy of your pancreas, of your skin, maybe your liver, and we test for hemosiderosis. This may indicate that you have an overload of iron. Loss of regulation. Let's say your hepcidin is not getting stimulated enough. This could be an issue. Another reason is ineffective erythropoiesis. Now, why is that? An ineffective erythropoiesis, again, what's erythropoiesis? It's just making new red blood cells. So if your body is struggling to make red blood cells, something called erythropoietin is going to increase. Erythropoietin is the protein that stimulates erythropoiesis, this process of making red blood cells, because it's trying to compensate, it's trying to make more red blood cells, but it's not working. So these increased levels of erythropoietin are actually going to suppress hepcidin, which is going to be causing iron overload because it's not regulating iron absorption. Too much iron is being absorbed. Another reason is repeated blood transfusions. This is self-explanatory. You have more blood, meaning you have more iron than usual. Now, what do we observe? Diagnostically, how do we know that someone might be experiencing iron overload? Well, two main ways. If they have an increase in total body iron stores, or an increase in hemosiderin or hemosiderosis. And how do we know this? If we biopsy the liver, the pancreas, the skin, or the heart, which is way harder to do. I think I'd just go with skin, pancreas, or liver. But the heart is also somewhere where hemosiderin can get accumulated nonetheless. If we biopsy it and we do a Prussian blue stain and it comes back positive, then hemosiderosis indicates iron overload. If we have increased total body iron stores, as you're going to have more Fenton reactions, forming free radicals that are going to be harmful to your healthy tissues. Again, this is why we need to be on the lookout for iron overload. Now, before we wrap up, point you could say that I want to make about regulation. So, ferritin synthesis and transferrin receptor 1 synthesis. This is the protein that is responsible for binding iron. This is the receptor that is responsible for releasing iron. So let's start with ferritin synthesis. So let's try to break this down. When do we need ferritin? What does it do? It binds iron, right? If increased iron levels, then we're going to need some ferritin to bind that iron. Meaning our body's going to be like, okay, so let's make some ferritin to deal with this extra iron that we've got. We always start with mRNA translation when we're talking about protein synthesis. There's a bunch of other steps, but that's irrelevant for now. But starting from here, this mRNA is going to be transported from the nucleus to the cytoplasm, where the light and heavy chains are going to be synthesized, forming ferritin, etc., etc. So let's say we've gotten to this point and we're synthesizing all this ferritin, and all the iron is dealt with, meaning iron levels are now lower. Once the iron levels are lower, this is when something called the iron regulatory protein steps in and binds to the iron response element. This protein binds to this element and blocks any further mRNA translation. In other words, blocks any further ferritin synthesis because we've got enough. And we know that we've got enough because we have lower iron. Now, on the other hand, transferrin receptor 1 synthesis, which this receptor releases iron. So we can say that when we have low levels of iron, is going to be the exact opposite. Low levels of iron is going to tell us that we need more transferrin receptor 1 to release iron so that we can get that level back up. So how does this happen? We actually need something to happen here. We need the same reaction here. We need this iron regulatory protein to bind to the iron response element in order for us to have transferrin receptor 1 so that we can have more iron be released. Now, let's say we got more iron to be released because transferrin receptor 1 has been synthesized, and iron levels are back up. Once iron levels are back up, then IRP does not bind to IRE anymore, stopping the synthesis of transferrin receptor 1. So over here is just a nice table that summarizes everything really nicely. Let's say we have increased iron in the plasma. Increased iron in the plasma is going to make sure that the iron regulatory protein does not bind to the iron response element. What does this do? Not binding these two elements is going to allow ferritin synthesis and stop TFR1 synthesis. Now, why do we need these two to happen? Because we have plasma, we need the ferritin to bind this iron, and we don't want any more transferrin receptor 1s because we don't need any more iron to be released. On the other hand, if we have lower iron in the plasma than usual, then IRP will bind to the iron response element. What does this do? This does the exact opposite because we need the exact opposite to happen. This stops ferritin synthesis because we don't need any ferritin, and it's going to allow TFR1 synthesis to release iron to get that level back up. Before we wrap up, there's a few points that were mentioned in the lecture slides that I want to bring your attention to. Over here, there's something that we didn't go over. So these 3 points, first of all, calcium impairs the absorption of all iron. You can remember this by, let's say, we have a bowl of spinach and a glass of milk. We also have a bowl of spinach and just like apple juice or something, or like orange juice. This is not an ideal meal because if you're going to be eating the spinach for its iron, then you're not going to be able to get all the benefits since calcium impairs the absorption of iron, which is why going with the orange juice is probably a better idea, even though orange juice is not really good for you. But that's for another time. So calcium impairs the absorption of all iron. Another thing, interleukin 6 induces hepcidin during inflammation. Interleukin 6 is a cytokine that is released during inflammation. Again, just reinforces the fact that we already covered, that hepcidin is stimulated during inflammation, which is why iron deficiency is associated with inflammation, because hepcidin is always stimulated and we're not getting enough iron absorption. Over here we have erythropoietin suppresses hepcidin during hypoxia. So again, we talked about this. Erythropoietin is the protein that stimulates red blood cell production. And let's say someone is undergoing hypoxia or ineffective erythropoiesis or just any condition that causes them to be low on oxygen overall. Their body's going to be trying to make red blood cells, and by doing that, they're going to be upping their EPO, their erythropoietin protein, to try and ramp up that red blood cell production. However, EPO, erythropoietin, suppresses hepcidin, meaning you have an increased chance of iron overload if you are in a oxygen compromised situation. Some more information: hepcidin is a glycoprotein and it is synthesized in the liver. It is induced when plasma iron levels are high. Its induction will result in reduced iron absorption, reduced macrophage iron recycling, hypopheremia, hypolow fer iron emia blood, reduced placental iron transfer during pregnancy. Over here is just the actual mechanics of how hepcidin is downregulated. So we have a gene called the HFE gene, codes for a protein that senses transferrin levels, or more specifically, transferrin ferric complex levels. Once these levels are too low, it's going to tell the hepcidin to go to the ferroportin on the macrophages and the enterocytes and degrade itself. And that's it. I hope this helped. Best of luck.